thank you for coming today. Since my Italian is not very good yet, so I will speak in English, but I will go slowly through my slides. And please, if something will be not clear, just let me know and I will try to explain it better. So one line of research which we are doing at IFOM is that we are studying genes which are involved in DNA repair. And today I will give you some overview about basically the field. Why is it important to study genes which are involved in DNA repair, and what's the relationship to cancer and aging? So the idea is not to go into detail, but more or less to give you overview. So first we will be talking about our DNA. So you will see that in our cells, are under the attack of various factors which can cause DNA damage. Then we will see that cells are smart and they evolved a mechanism to repair that DNA damage. Then we will see how important is DNA repair for our health and what is happening if DNA repair is not working properly. And we will also see how understanding of DNA repair mechanism is providing new opportunities for disease detection and also for treatment. So we can start from more general things and then we can go into some details. So we know that as we are sitting here, our body consists from different systems. So here we have, for example, digestive system, and the digestive system consists from various organs. So here we have, for example, stomach, intestine, and different organs consist from different tissue. Tissue are built out of cells. So cell is basically basic unit of our body, and in cells we can find various molecules such as DNA, which are made out of atoms. So today basically we will be focused mainly on cellular level and chemical level, such as molecules, DNA and proteins. So we know that human cells represent basic structure and functional unit of our body. Each person, maybe you remember that each person contains more than 37 trillions of cells. Our cells have various shapes and sizes. Average size of cell in our body is between 10 to 100 micrometers. 100 micrometers is 0.1 millimeters, so they are very small. And in our body we can find around 200 different cell types. These different cell types, they have different functions, and they are also localized in different organs. So they have different size, different shape. And the aim is basically to proceed with different functions. So here we have example how real human cells look like. So here we have cells which we are using for our research. So here are cells which were isolated from the bone of a 15 years old girl which suffered with osteosarcoma. Osteosarcoma is a tumor which can be found in bones. And those cells were isolated from that girl in 1964. Since we are talking about cancer cells, these cells are basically immortal. So since that year, basically scientists keep them in cell culture. So those cells are able to grow on plastic dishes. Inside the dish we have a medium. It looks like blood, but it's basically water with nutrients. And other chemicals which are basically simulating human body conditions which we can find in our body. So here we have many cells. You can see that this is cell, this is cell. Here we have scale bar. So this wide line is 100 micrometers. So you can get some idea about the size of the cells. And here we have two cells from this photo basically in detail under the microscope. So that's the real shape of the cells. And as I told you, they can grow on plastic dishes, and these plastic dishes we then keep in a cell incubator, which is a device similar to the fridge, but instead cooling down, we keep them at 37 degrees under certain humidity and CO2 level. The purpose is basically to mimic conditions in our body. So here we have a movie showing how those cells, which you saw on previous slide, basically look in real life. So here we will see cells on plastic dish, and you can see that cells are independent and they can move around the dish. So this is one cell, this is another one. And those cells basically you can find in our body in bones. So it's important to understand that basically our body consists from cells like we can see here, and that they are pretty independent. Okay. So here just some information just to remember, probably you know that from school about cell structure. We will go not into detail. So here we have model of the cell. So you know that on the top there is plasma membrane. Below you can find cytoplasm with various organelles which perform different functions. And in the middle of the cell there is nucleus which contains DNA. So this is model, this is again a real cell. So cytoplasm is here, which is on top of the cell. Below we can see plasma membrane. And nucleus under the microscope looks like this. So here is nucleus containing DNA. So some information about human DNA. So here is cell, here we have nucleus. As I told you, DNA is inside the nucleus. It can have different levels of compaction. So DNA can be in form of chromosomes, or it can be just as fiber. So human DNA has a double-stranded helical structure, which we can see here. And the diameter of human DNA is 2 nanometers. That means that human DNA, which is in our cells, is 50,000 times thinner than human hair. Each cell contains 2 meters of DNA. So it's basically fiber, which has length 2 meters. And since we have 37 trillions of cells, so the total length of all DNA in our body is 6 times the distance of Moon from the Earth. So you can imagine how much DNA our body contains. And since DNA is 2 meters and the nucleus is very tiny, so then DNA is highly compacted into cell nucleus. And if we compare it with situation in real life, it's basically like compacting rope, which is long as a football field, down to the ball of 1 centimeter in diameter. Here we have again a 3D model of DNA, so we can see double helical structure. And here we have real DNA from our experiments. Basically, this DNA was isolated from the same cells as you saw in the previous case. So you can really see that it's a fiber. This is quite a difficult experiment. I think there are just few places in the world where you can do this experiment, and this institute is one of them. And here, to see DNA, we are using electron microscope with magnification 250,000 times. So now we are going to talk about DNA and how DNA can be damaged. So I told you that we have around 2 meters of DNA in each cell, and this DNA can be damaged in our everyday life. So every day, basically, each cell receives more than 70,000 of DNA lesions per day. So it means in one day, basically, each 2 meters of DNA can be damaged more than 70,000 times. We know about some factors which are basically causing damage to our DNA. So one of them is, for example, sunlight. So you know that if you go for holidays and you spend some time on sunshine, so you can get red, and in a few days your skin starts to peel off. So it means that you basically killed your cells by strong sunlight, because these cells were damaged. So strong sunlight, if you go on sun without any protection, as sun cream, can cause more than 100,000 lesions per 1 hour. So that's higher than 70,000 per whole day. Another factor which is causing DNA damage is smoking, since we know that in cigarette smoke there are chemicals which can bind basically to our DNA and cause damage. Also, alcohol consumption is causing damage to our DNA, since during alcohol, once alcohol is metabolically processed, chemicals are released and those can bind to our DNA. Another factor is, for example, food. We can mention meat, which was prepared on open fire, such as steaks. If you let them burn down, then again chemicals can be present and they can damage our DNA. We can also mention radioactivity, such as radioactive gas. Radioactive gas called radon can enter basically from ground to our houses. And for example, in USA, 21,000 of Americans die every year because of presence of radon in their house. And then we can mention car emissions and emissions from industry, for example, from factories. So why is it important and why that's the problem? So you know from the school that DNA is repository of genetic information. So there is genetic information, and if this genetic information is basically damaged by all those factors, then basically DNA information is changed. So you know that the main function of DNA is the long-term storage of information. DNA is divided into short segments which are called genes. So we have approximately 25,000 genes in our DNA, and each gene serves as instruction on how to build a molecule called protein. I will go through this figure. So here we have cell. Inside the cell is nucleus. Inside we have DNA. And then DNA can be divided into 25,000 segments called genes. And each gene serves as information how to create protein, which is another type of molecules. So here is just some characteristic of proteins. So proteins are molecules which are doing the most of the work in our cells. So they are very important. We have many categories of proteins. So we have proteins which, for example, provide structure and support for cells. Then we have proteins which are responsible that cells can move, as you saw on previous slide. Then we have then we have proteins which works as enzymes, so they carry out chemical reactions in the cell. Cells can also transmit signal to coordinate biological processes. And other proteins, for example, can fight with foreign particles such as viruses and bacteria. Today we will be talking about another group of proteins, which are proteins which are involved in repair of our DNA. So cells are smart. They know that it's a problem once they get damaged their DNA. That's why inside the cell we can find a group of proteins which are involved in repair of that DNA. Just to give you some idea about complexity of our cells, so here we have basically one cell, here we have nucleus, and all these dots represent different proteins. So as I told you, in our cells there is around 25,000 different molecules, different proteins, which have interactions between themselves, and they all basically proceed with different functions. So imagine that here we have basically around maybe 100, 200 proteins, but in a real cell there will be around 25,000 proteins, and all of them will be presented in many millions of copies. So it's a very complex system. So now we will be talking about DNA damage. So here we have DNA, and you saw that we have different sources of DNA damage. So and those different sources of DNA damage can cause different type of damage. So for example, cigarette smoke will break one fiber of your double helix. Then, for example, radioactive gas, radioactivity can cause a double-strand break. So basically, DNA will be broken completely. So different sources of DNA damage can cause different type of DNA damage. And then we have proteins which are able to repair these DNA breaks. So we have, so far, we know about 7 molecular pathways which are able to repair DNA damage. You can imagine, we don't need to go into details and know names. It's just to show you that those proteins, basically, they work in groups. You can imagine it as some small teams of proteins which will come once they will see this damage. So if there is single-stranded DNA caused by, for example, cigarette smoke, then this team will come and repair it. If there is different type of DNA damage, then other proteins will come and repair it. So how is DNA damage in our cells repaired? So we can go just into 3 very easy steps. So you can imagine here that we have DNA, and here is problem. There is a DNA break. So in that case, proteins basically need to perform 3 steps. First, they need, of course, to find the problem. They need to find where is DNA damaged. So we have proteins which works as a sensor, so they are going all around the cell, and they are basically screening for those kind of problems. Once they will find it, they basically localize on that place, and they are able to signal the presence of DNA damage. So those proteins will inform other proteins in the cell that there is problem, that there is broken DNA. And as a third step, another kind of proteins will come, and they will basically repair it. So here, we will not go into detail. Here is just one current model. So that's how we think that it works. So this is pathway called nucleotide excision repair, which is repairing basically specific type of DNA damage. So we can see that there are many different proteins. Here we have DNA. So those proteins, they basically will come, and through several different processes will repair DNA. Here we have just a very short movie to show you basically what's the model, how it works. So there you will see one protein which will be cutting DNA, and another 2 proteins which will be basically fixing DNA. What we see here is, here we are inside the cell, and here is cell nucleus, and inside cell nucleus there is DNA. Okay, so now we are going into the cell nucleus. And here we have chromosomes, so that's DNA. Here is double helix. And basically this is one of the proteins. So that's molecule which has some function in our cell. This protein is called Cas9, but here we can see DNA. Here is protein basically which can sit on DNA, and now it can cut DNA. Because in some situations it might be also important for cell to cut DNA. So now you can see that DNA is cut it. And here we have double-strand breaks. So both helices are destroyed. So then other proteins basically will come and they will fix this DNA damage. So you see that is very important for us that our DNA is basically intact, that is not damaged. So here is a slide showing why we need to understand how cells work. So you saw already that cells are very complicated. It's like small machines, and they can get broken. So basically human diseases are caused by dysfunction at either cellular level or molecular level. So cellular level, it means that you basically damage all your cells if you get broken your leg, or for example if you burn your skin. But then we have also problem at molecular level. So that means that some of the proteins which are inside the cell, they don't work properly. So I told you that there are 25,000 proteins in million of copies, and they are basically making some teams which are performing specific functions. So now if you imagine that one of the protein is not working properly because of some mutation in DNA, then you can imagine that it can have impact basically on a whole cell, and then of course on our health. So why we need to study basically how it works, what's the function of all molecules. You can imagine it basically with car. So if you want to go to school or to work, and you got this nice car, this Ferrari, so what can happen that you will get out of your house and you will see that there is problem with your wheel, that you have flat tires. So since you understand what is this and how it works, what different parts of cars are for, so then of course you will just fix your wheel. You will not open engine and you will not start search for problem inside the car. That can be quite expensive. So since you know what these parts are for, then you fix just wheel and you go to work. The same is basically with cells. So here we have cell, and also inside the cell we have different basically parts, different organelles, and inside organelles we have also different proteins. So for us, if we want to treat human diseases, it's very important to understand what for are all these parts, what's their function, and basically what it means if some of them are not working properly. So deep knowledge about ourselves is necessary for understanding what happens when we become sick and how to treat the disease. Here I put just one example basically of experiment which we can do here, for example. So if we are interested to test if some protein, we found for example new protein, and we want to test if this protein is involved in DNA repair, if its function is basically to come and repair DNA. So then we can use a system called green fluorescent protein. So it means proteins in our cells, they don't have any color, but we can basically make this protein which we want to study green. So it means we take protein and we add small fluorescent mark. In this case it will be green fluorescent protein. It's like if you want to go for running, so you want to come back and see where did you go and what was the speed, how many kilometers did you run. So for us also, for our research, is important to know where this protein is going inside the cell, what it's doing and when. So through this GFP, green fluorescent protein, we can tag this protein. I will show you more details. So here we have information about green fluorescent protein. So you probably know that jellyfish, it's here, is organism which lives in the cold water of Pacific Oceans. And of course this organism has DNA, and there is gene which is coding green fluorescent protein. And this green fluorescent protein is light activated glowing green protein. So it means if we shed light on this animal, it will turn green. So what scientists did, they basically took this part of DNA, they took gene from jellyfish, and we can insert it into various organisms. So this gene was inserted into yeast, roundworm, fruit fly, rabbit, and mouse. So for example here we can see mouse. So these three in the middle, they are normal. Those three mice are basically expressing gene from jellyfish. So why we are doing it is not just to create something interesting. Basically this approach will allow us, as I told you, to track protein. So here we have a microscope which is equipped with camera and with laser. Everything is connected to computer. And then what we can do is that we can take cell, and by laser we can make basically damage inside the cell. So here will be basically part of the cell which was irradiated with laser, and here we have nucleus, so DNA is stained in blue. And then here we have video, and this is again nucleus, and in green is a protein which we want to study. So its protein is in nucleus and is everywhere, since this one protein is present in millions of copies. So here we have situation before irradiation with laser. And And you can see that protein is basically diffused everywhere. So that's one of the proteins which is basically scanning if there is any DNA damage. And now I will show you what will happen if we perform with laser and we make basically a line inside the cell where DNA is damaged. So what we will see basically will be that this protein will recruit. It will find place where DNA is damaged and it will go there. You see, so all this is basically protein which was previously, it was everywhere, now is on the place where DNA damage is. So that tells us basically that this protein has some function which is related to DNA repair, because that's why this protein is attracted to the place where DNA damage is. So you saw that we have DNA can be damaged, and then that we have proteins which are able to repair that DNA damage. So then we have basically proteins which are able to repair problems with DNA. So now what is happening if some of those genes is not working properly? So if DNA repair genes are not working properly, it can lead to serious diseases. So today we will be talking shortly about cancer. We will be talking about hereditary diseases with defects in DNA repair, and also about premature aging syndromes. So here we have just very basic information about cancer. So what is cancer? Cancer is basically when abnormal cells divide in uncontrolled way. So here we can see normal cell, and here we have cancer cells. So what was happening basically that in DNA there was some mutation, some defect, and protein which is supposed to be created based on this information in your DNA, basically because DNA is damaged, then protein is not functional. And that can lead to the state where basically cells are dividing without any control. So those cells are growing much faster than a normal cell. Can you imagine that, for example, from a normal cell, if you are healthy, in 24 hours from one you will get 2 cells. However, cancer cells, they can proliferate, they can divide much faster. So instead 2 cells in 24 hours, you will get 4 cells, for example. It's important to mention that cancer is not just one disease, it's a whole collection of different diseases. So we have around 200 different types of cancer, but basically problem is very similar. So it's uncontrolled proliferation of cells. We know that the cells can cause tumors, which is state when cancer cells are growing through normal tissue. We also know that there is some primer tumor. So primer tumor is the name for where cancer starts. But these cancer cells, they can also go around our body, and they can sit in other areas. So in that case, we are talking about secondary tumors or metastasis. Here we have some numbers about cancer. So here are data from 2012. So in 2012, around 14 million of people got cancer. It's worldwide data. And about 8 million of people died. Lifetime risk, so basically here are data from United Kingdom. So 1 in 2 people born after 1916 United Kingdom will be diagnosed with some form of cancer. So you can see there is a problem which is concerning all of us. And then what's the risk for cancer in men and women? So risk in men is males have the highest lifetime risk of prostate, lung and bowel cancer. And in women, females have the highest lifetime risk of breast, lung and bowel cancer. Here we have a chart showing that age is basically, that incidence of cancer is strongly related to age. So here we have different age, and here we have number of cases when cancer was diagnosed in particular age categories. So we can see that when we are young, so there is not so high risk of cancer. However, with prolonged age, basically incidence of cancer is much higher. One explanation is, again, it's concerning our DNA. So if we are longer, we have basically longer time to accumulate mutations, to accumulate basically problems in our DNA. Our cells are not working properly if we are longer, if we are older, and that can lead to cancer. Here we have factors which are increasing risk of cancer. Again, it's correlated with factors which are damaging our DNA. So first, basically, cancer risk factor is the smoke from tobacco. So tobacco smoke is responsible for 33% of all cancers. Second factor is obesity. So if we are not keeping normal weight, so then it can lead to 20% of all cancers. Then we have other factors which are pathogens. So here we can mention viruses. So it doesn't mean that cancer is transmitted from person to person through virus, but it means that there are viruses which can cause infections, such as we can mention hepatitis C, which is virus which can infect our livers. And during its life cycles, basically this virus is entering into our cells and it's inserted into our DNA. And once this virus inserts into our DNA, what can happen that some gene important for us will be disrupted, will be basically damaged. Other factors are, for example, lack of physical activity, of course eating food which is not healthy, then alcohol and sunlight. So basically overall 42% of all cancers can be prevented if we will keep some kind of healthy lifestyle. So now, how cancer basically can arise. So as we said before, cancer is a genetic disease. So here we have normal cell, and here we have cancer cell. In the middle is precancerous cell, which already acquired some problems, but it's not cancer cell yet. So if we imagine normal cell, as we said before, so there are many factors which can damage our DNA, such as sunlight or pollution from industry. And these factors can damage our DNA. As we said before, we have proteins which can fix it. But sometimes what can happen is that those proteins which are supposed to repair our DNA are not working properly, because maybe they are disrupted by some virus, or they are just this DNA damage disrupt gene, part of DNA where some important protein for repair is localized. And in that case, DNA damage starts to accumulate. And once there is some level of DNA damage, then cells basically cannot longer understand the information which is stored in DNA. So then instead making proper proteins, they make different mutants. And that can basically lead to cancer. So cells with damaged DNA can start to multiply without any control. I told you that they can grow faster. And basically then cancer will arise. Here is just one slide. We will not go into detail, but just to show you how cancer is basically a complex problem. So here we have information that different DNA repair proteins are defective in various cancer. So we are not going into detail, just imagine that, for example, here we have breast cancer, and here we have ovarian cancer. And all these are proteins which the main function is to repair DNA. However, for example, in breast cancer, different proteins will be not working than in ovarian cancer. So in other words, basically in breast cancer you can have different mutations in genes which are repairing DNA than in ovarian cancer. So based on this, you can see that it's really not one disease, that it's many various diseases. Here we have a slide showing how our understanding of DNA repair mechanism can provide new opportunities for cancer treatment. So here we will be talking just about breast cancer. So Here basically we have standard therapy, here we have personalized therapy. So that's where we are mainly, that's where we want to get. So if you imagine that here we have 50 people and all those people, they have breast cancer. So if we are looking on them, if we are doing diagnosis, we see that they have breast cancer. But what we need to do is to find out basically which protein is not working. So let's say all those people, they have 20 different proteins which are supposed to repair DNA. And all of them, they have breast cancer. But you can imagine that, for example, in person which is marked in yellow, so protein marked in yellow is not working properly. If we are looking person in red, so this person has also breast cancer, so it looks the same. However, problem is different. Protein which is basically not working properly is different than in yellow person. Again, blue person, so again breast cancer, but different problem. And then if you imagine that basically we say, okay, so we have group of people with breast cancer and we don't care about this, so we give them just one drug. So we give them, let's say, drug A, blue drug. So then what will happen if all of them will get the same cure is that basically this drug A will be working only for people who are blue, who has basically mutation in this particular gene. And since we don't know what's the other mutations, what's the problem of other people, yellow, green and red, then basically those people will not survive our therapy. So we are not going to help them. So what we want to do, basically what is necessary to do is to study molecular mechanism and not just say that those people, they have breast cancer, but it's important to know why they have breast cancer. So in that case, this group of 50 people, it can be divided into small subgroups, and we know that people in blue, they have basically the same problem. So they have mutation in this protein. People in yellow, they have also the same problem, but different than people in different colors. So then basically we can provide those people with 4 different drugs, and all of them will be cured. So that's why basically we need to study molecular mechanism, which is behind cancer. So just a few last slides. So now we will be talking about hereditary diseases with defects in DNA repair. So it sounds complicated, but it basically means that these are genetic diseases. So they are arising through problem in our DNA. They are hereditary. That means that defective gene is basically passed down from parents to child. So parents, they have defects, some gene is mutated, is not working properly, so then they can pass this piece of DNA to their child. And basically all these syndromes, they have the same problem. They have defect in DNA repair genes, what means that patients basically have impaired repair of their DNA. So we have several syndromes known already. I will show you just this one, which is called xeroderma pigmentosum, just to illustrate and have some example. So xeroderma pigmentosum is basically a disease which is caused by defects in genes involved in repair of DNA damage caused by sunlight. So as we said before, sunlight can cause damage to our DNA, and we have proteins which are supposed to repair that DNA damage. However, in those people, basically some of those genes which are supposed to repair damage after being exposed to sunlight are not working properly. Incidence of this disease is 1 to 1 million. That means that one child out of 1 million will get this disease. And here we have symptoms. So this disease is diagnosed usually around age 1 or 2 years. And those people are extremely sensitive to sunlight, since they don't have genes working which can repair damage once they are exposed to sunlight. They are extremely sensitive. So it basically means, as we can see here on that kit, that if they go outside without protection, without clothes or without sun cream or sunglasses, they basically, after a few minutes, their skin gets burned. So those kids are also called moon children, since they can go outside basically without sunlight protection only during the night. And they have increased risk of skin cancer. So skin cancer appears by age 10. And beside these problems, also around 30% of patients, they have a problem which involves nervous system, such as they have problems with coordination, they have problems with walking, and they are also losing their intellectual functions. Various length of life is different here, and basically depends if they get, beside these problems with sensitivity to sunlight, also problems with nervous system. And here in the last part, just the last 3 slides, basically we'll be talking about premature aging syndromes, since defects in DNA repair genes are not basically linked only to cancer or problem with DNA repair, but also it concerns premature aging. So here we have DNA damage theory of aging, saying that aging basically is a consequence of accumulation of DNA damage in our cells. So it's a complex problem, but just to illustrate, basically we can imagine that if we have properly working DNA repair, or even better working, then we can live longer. However, if we have some defects in DNA repair, that allows basically accumulate mutations in our DNA, and that can lead to premature aging. So we know that there are several premature aging syndromes. So premature aging syndromes represent group of rare genetic diseases which can mimic physiological aging. So basically, in fact, it means that those patients, they are aging faster. So they have certain age, but they look older than they are. And individuals with those diseases, they have a shorter life. And again, as we said, one of the main causes of aging syndromes are defects in genes involved in DNA repair. Again, we know already that there are several syndromes which were found, but I will show you just one, which is Hutchinson-Gilford progeria. So in this case, basically, we are talking about defect in a gene which is called lamin. And this gene basically is coding protein, which is responsible for regulating shape of our cells. And this protein is also involved in DNA repair. Incidence of this disease is 1 to 4 million. So it means that 1 child out of 4 million will get this disease. And here we have symptoms. So those kids, they look basically normal when they are born. However, around 1 or 2 years of age, they will start to age, and they age 10 times faster than other population. So we can see here how they look. So they have basically growth delays. They have a small face in comparison to the head. They have prominent eyes, hair loss, aged looking skin. They are also losing fat under the skin. And interestingly, they can also get heart attack at very young age. So they are suffering by heart attacks around age at 13. However, intellectual development is not affected. And those patients, they live to about 15 to 20 years. And the main cause of death is heart attack. So I think this is a nice illustration. If you imagine that we have 25,000 genes, and if one of them is not working properly, in this case lamin, how it can basically change our body and basically quality of life. So here we have some conclusions. So as we saw, we didn't go into detail. It's more or less just that you have some overview about field which we are studying here and why it's important to understand it and study this. So we saw the DNA in our cells is being damaged every day, and cells, they have basically mechanism how to repair that DNA damage, since they know that it's important to keep integrity of our DNA intact. We also saw that functional DNA repair is crucial for prevention of human diseases such as cancer. And our understanding of DNA repair mechanism is providing basically new opportunities for disease detection and for treatment. That's basically our aim. Last conclusion, I think that you saw that our cells and our body is a very complex system, and I can tell you that a lot of work still needs to be done. Here we have some data again about cancer, showing basically that cancer survival in United Kingdom has doubled in the last 40 years. So it means that 40 years ago only 24% of patients basically survive once they got cancer. Now around 50% of patients can survive. But there is a huge variation. It depends on what kind of cancer people get. So there are cancers which can be cured, basically from, let's say, 98% patients will survive. But then we have also cancer where only 1% can survive. So a lot of things need to be done. So if you find this topic interesting, so you can consider some scientific career. It's not just for biologists or molecular biologists, genetics. We have here also many people who are mathematicians, physicians, who basically work on computers, since we are processing a lot of data and someone needs to analyze all data. Okay, thank you very much for your attention.